All right, so today we're going to talk about enzymes, but before we jump into enzymes, always a quick little review because it's super important to always go back to what we've already learned. So we talked about macromolecules on Monday and Tuesday. How many are there? 4. What are they?
Lipids, protein, carbohydrates, and nucleic acids.
All right. Carbohydrates, proteins, lipids, and nucleic acids. The next thing that we had to know were their monomers. First of all, what's another name for a monomer? Subunit or a building block. That's right. Okay. So what is the monomer of a carbohydrate? Monosaccharide. Nicely done, Gary. Okay. So mono means what? One, and saccharide means sugar. So one sugar or a simple sugar. What's the building block of a lipid? Yes, very good. A glycerol head and 3 fatty acid chains. That's right. Okay, so glycerol head, 3 fatty acid chains. What's the building block or monomer of a protein? Amino acids. That's right. And about how many are there? 20. That's right. Next, we need to know the building block of a nucleic acid. Nucleotide. That's right. Who remembers the 3 things that make up a nucleotide?
Nitrogenous base.
Nitrogenous base, yeah.
5 carbon sugar.
5 carbon sugar and a phosphate group. That's right.
When did we learn this?
Monday and Tuesday.
I know we learned that, but when did we learn all these little other things? Like I saw the video and all that stuff.
Monday and Tuesday.
I swear I was listening.
Next, we're going to move on to functions. So what are the 2 functions of a carbohydrate? Yep, quick energy. And structure. Very good. Okay. Next, we need to know the function of a lipid. There's quite a few. Insulation. Cushion. Stores energy. What does it make up?
Waxes.
It does all of those, but what in particular does it make up? It's part of the what? Something that I told you we'll talk about coming up. Found on the outside of a cell. Cell membrane.
Yeah.
Okay, makes up the cell membrane.
You know,
That's right.
Javier.
Next, we need to know the functions of a protein, but before you tell me those, what was the one thing I told you? Proteins.
Most versatile.
Very good. Most versatile. So give me some functions of a protein.
Build muscle.
Build muscle. Build muscle. That's good. All right. Transport substances.
Store substances. No, that's what it is. No, store DNA.
No.
RNA.
No.
Achoo!
Fight disease.
Fights disease. And it does what?
It's a speed reaction.
What about, yep, speed up reactions. Okay, speeds up chemical reactions. And what's that called? An enzyme, and that's what we're going to talk about today. And then last is nucleic acids, and what do nucleic acids do?
Transmit and store.
Yep, hereditary info. Store and transmit hereditary info. Which one does the storing?
Yeah.
And which one does the transmitting? Why?
Because DNA is too big.
Too big to do what?
Leave
To leave the nucleus. All right. The last thing, we're going to go over some examples. So let's go backwards because you already gave me nucleic acids. What are the only 2 examples? DNA and RNA. What are some examples of protein? Chicken, eggs.
Nuts.
They are, but what else? Okay, very good. What else? You told me it. What speeds up reactions?
Lactose.
No, it's the powder.
Now, what speeds up reactions that we're going to talk about today?
Enzyme.
Enzymes. Okay? And then what else? Tell me the 2 biological examples I told you to remember. Insulin and hemoglobin, right?
Wait,
Very
is
good.
lactose a protein?
No, baby.
What is it?
Lactose
It's
is
a
the
carbohydrate.
sugar, it's a carbohydrate. Alright, next, give me some examples of a lipid.
Blubber.
Blubber, wax, oil, any kind of fat, right?
Grease.
And yesterday we also talked about grease is a good one, and we talked about what? The medication I said,
Steroids.
steroids, right? Alright, and then last is carbohydrates, the list can go on and on and on and on.
Bread.
Bread, starch,
Lactose.
pasta, lactose, glucose, fruit, sucrose, vegetables. What else?
High fructose corn syrup.
Girl, you crack me up.
Someone said fructose.
Yes,
fructose.
fructose.
So is that right?
What else? How about some fruits? What's found in a plant that we talked about? Cellulose, right? What do we find in pasta and potatoes? Starch. Is that simple or complex? There we go. Boom, boom, boom. All right. So, does anybody have questions about that? All right. Last thing before we jump into enzymes. What elements make up carbohydrates?
Carbon,
Carbon, hydrogen, oxygen. What makes up a lipid?
Carbon, hydrogen, oxygen.
Same thing.
No.
Yes, if you remember in the order I taught you, cho cho chon chon's the way to go. So, also carbon, hydrogen, oxygen. What makes up protein?
Amino acids.
And nitrogen.
Now we add nitrogen, and then what's the last thing we add for nucleic acid?
Phosphorus.
Phosphorus. Because of those phosphate groups. Okay? Any questions? We're good? All right, so today we're going to talk about enzymes in specific. So chemical reactions change one substance to another. So up here, for example, we have 2H2 plus O2, which yields 2H2O. What's H2O? Water. Okay? So you've got hydrogen combining with oxygen, and now they are making a water molecule. So when you see a chemical reaction, an equation, there is always an arrow. And that arrow is the most important part of the reaction. And the reason is because that's what's telling us what's being made. So essentially, we are saying what it's making or yielding. Okay? So hydrogen and oxygen are making water, H2O. So this is important to know. These are going to come back later as well. So reactants, also known as substrates, and substrates was one of your vocab words this week, those are what come before the arrow. So those are what we put into a reaction. And I'm going to give you an example of this to help you remember. What comes after the arrow is known as our product. So that is what is being made. Okay, so that's what we end up with. So a good example is if we were, do I have any bakers in here? None of my boys bake in here? All right. So if you were to bake a cake, right? What do we need to bake a cake? Let's just say, let me make it easy. Those are all correct. But you went to the store and you bought a box of cake mix. Okay, water. So you might need the cake mix you bought from the store, some water maybe, or oil. What else? Not yet. What else? What might be cracks? Some eggs, okay? And then the cake mix already has all the other stuff that we need, all the dry ingredients, right? So we're pretty much just putting the wet ingredients in. So all of those ingredients that we need to bake the cake would be known as what? The reactants or substrate, okay? So when we mix those all together and we put them into the oven, what do we get at the end? The product, the cake. We get the cake, okay? and the cake is our product. Does that make sense?
Yep.
All right, remember that because we're going to talk about it as we move on. So, I want us, obviously we're not circling things, but looking up here, I want you to tell me for number 1 what the product is. What's the product for number 1? NH4Cl. Why is that the product? Because it comes after, okay? Those boxes should actually be arrows, but they're not showing up because I went from a PowerPoint to a Google Doc, or a Google Slides. All right, for number 4, what's the product?
C and CO2.
4.
H2.
What's the product for number 4?
I didn't know.
Oh, number 4 is sunlight.
Glucose.
Glucose plus
And
Glucose plus O2, because it's what comes after. All right, so now let's look at number 2. What are the products?
H2 and CO2.
Okay. For number 3, what are the substrates? Glucose and oxygen, because they come where? Before. So does everybody understand that? Okay. All right. This is another example of a reaction. What do you see up there? A match that's already lit. Okay. So how is that a reaction? Yes. So you strike it against the matchbox. So you would take your box of matches, right? You take the match out of the box and you strike it against a specific side, right, of the matchbox. So what are my substrates in that reaction?
The specific side.
Yep, the specific side of it. Look at you go. All right. And the match. What's the product?
The fire.
The fire. Okay, that's the product. All right. So chemical reactions require energy to get started, and we saw this yesterday and got goofed up a little bit. Okay, this was also another one of our vocab terms. So the energy that's required to start a reaction is known as the activation energy. The best way to remember that is that start also means to activate. So activation energy is what we need to start the reaction. Once the activation energy is supplied, then the reaction can happen. Okay? So on the left side, you see your reactants or substrates going in. Here's our activation energy that we need. And then once this happens and the reaction occurs, we get our final product. All right. So super important. An enzyme is known as a protein catalyst. And a catalyst is something that speeds up a chemical reaction. Okay? And it does this by lowering the activation energy that we need to get it started. So if you look at the two pictures on the board, okay, one of them has an enzyme and one of them does not. Obviously it tells you, but if it did not tell you, and you just looked at those pictures, which one would have the enzyme and which one would not? The bigger peak or the lower? The lower would have the enzyme because we were able, okay, to have less activation energy. The one that doesn't have it requires much more.
Wait, I have a question.
Yeah.
So, you said an enzyme is considered a protein activist?
Protein catalyst.
Oh. Catalyst.
C-A-T-A-L-Y-S-T. Catalyst. Okay? Ricky, what are you typing away? I'm up here teaching. I know, but that's not what this is for. That's why we're face to face. Okay? All right. So enzymes are protein catalysts. So that means that they are a protein. That's the macromolecule that they fall under. So we need enzymes for every process that happens in our bodies. So for example, we need them for digesting food and even moving around. So remember I told you, I asked yesterday if anybody was lactose intolerant. Did I ask that in here? Okay. So if you are lactose intolerant or know somebody that is, that means that we, or whoever is lactose intolerant, is lacking the enzyme lactase. So if you don't have that enzyme, you are unable to break down the sugar lactose. So that is why you are lactose intolerant. But they do make pills that you can take, okay? And that helps break that sugar down. So we already said this, what biomolecule are enzymes? Proteins, yes. And what do they do? Speed up reactions by lowering the activation energy. That's right. Boom. All right. So the next thing we need to know is this diagram. Okay. So the substrate is the material that an enzyme binds to, which is also known as our reactant. That was another vocab word. The complex is when the enzyme binds to the substrate. So it is possible that you would see something like this. Okay. So here's your substrate. Here's your enzyme. When they come together, that makes your complex, which is known as the enzyme substrate complex. And at the end, we get our what? Our product. Remember back to bacon and cake. All right, so now looking up here, which one's the substrate? Tell me colors and be specific. Yep, okay, the green and blue, purple, the circle and square, the very first one. That's the substrate because it's going where? Into, okay? Think of before the arrow. Which one's the enzyme? Which one's the enzyme? Be specific with colors.
The green, the orange.
The orange. Just the orange. The orange is the enzyme. And then which one's the product?
And green circle and purple.
Okay, so at the end, there will come at the end, that is the substrate essentially, in this case, broken apart. Okay? So, substrate, enzyme. They come together, this makes the complex. And then at the end, they come apart, and that is your product. But what's still there? The enzyme. The enzyme is still there. Keep that in mind. All right, here's another one. Let's try this again. Which one is the product?
Oh.
When they come apart.
Yeah. When they connect.
Just tell me colors here.
Oh,
Just-
purple.
The purple. The purple. So now this is opposite the last one you just saw, because we had one become what? Split apart. Now we're doing the opposite. Okay, so which one is the enzyme? The big pink one, right? Okay, and then which one is the substrate? The blue and the small pink, right? Very good. Everybody with me so far? Is it making sense as we go on here? All right. Last one. Which one's the substrate? Red and orange. Okay, in the first one. Which one's the enzyme? The blue. And which one is the product? The green. Okay, so know that when we go through a reaction, things can either break apart or come together. Okay. All right, so something else we need to know. Enzymes often end in ACE, A-S-E. Do not confuse this with carbohydrates that end in what? OS, O-S-E, okay? So right here, here's my example I just told you, lactase, which breaks down the sugar, lactose. Okay, another one is helicase, and you're gonna see this later when we get into DNA. This is what actually unzips DNA. There is amylase, which breaks down the sugar amylose. And last, there is lipase, which breaks down a lipid. The list of enzymes can go on and on. These are just some specific examples. All right, so what's a substrate? Tell me another name for it. Reactant. Okay, so it binds to the what?
The
The enzyme, right. What do they end in?
ASE.
ASE is lactase, an enzyme or a substrate. Enzyme, how do you know? Ends in ASE. How do you predict, or what do you predict is the substrate for the enzyme sucrase? Think back to this slide. So what do you think is the substrate for sucrase? Sucrose. That's it. Okay, that's the sugar, sucrose. So sucrase will break down sucrose. Okay, sometimes you gotta do a little bit of thinking. All right, so there are two characteristics of enzymes that we need to know. The first is that they are specific. So the best way to remember this is that they work like a lock and a key. So for example, if I have two keys on my keychain and I'm trying to get in this door and one says 305 and one says 311 and I put the 311 key in the door, am I getting in? No. If I put the 305 key in the 305 door, am I getting in? Yes. Okay. All right. So enzymes are specific. So they work with one substrate and they work like a lock and a key. Something else you should know is that when they come together, this on the outside here, that is known as the active site, and that is where they come together. Okay? The next thing, we need to know that they are reusable. What does it mean to reuse something? You can use it again. Okay? So remember back to those couple pictures? What happened to the enzyme? Did it ever change? No. Okay? So therefore, nothing happens to the enzyme, and the enzyme can be reused. So they're not changed, they can be used again. The substrate is what changes. So think of it this way. Back to my baking a cake. Where do you put the ingredients to mix them together? In a bowl. Does the bowl ever change?
No.
Do the ingredients change?
No.
Yes, what do they become? Which is what? The cake. You said product, you said cake. Okay? So you put all those ingredients in a bowl and you mix them together and then you put them in the oven and now you have a cake. Is that cake ever going to become those ingredients again? No. No. So that's why that is the substrate going in and the product coming out. Whereas that bowl, okay, or even whatever you bake it in, that's not changing, right? So that would be your enzyme. Does that make sense? Okay. All right. So what does it mean to say that enzymes are specific? Like a lock and key. Okay, they have to fit perfect. And what does it mean to say that enzymes are reusable? They can be used again. Okay? So up here, which one of these is my enzyme? Yep, lactase, that's right. Which one is my substrate or substrates? Lactose and water, H2O, and where is my product? Glucose and galactose. Okay, nicely done. All right, let's look up here. Which structure, or structure X is what? The substrate. And structure Z and Y, the enzyme. How do you know? Because the enzyme doesn't change. That's it. Okay? Any questions about that? I know it's a lot. We got a little bit more to do, but does anybody have questions? No? Another way to remember this, and if you watch the Ed Puzzle, you might see it in there because I think the Amoeba sisters talk about it, but has anybody ever heard of the game Pac-Man? Yeah? So Pac-Man? Chomp, 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 chomp. What does he eat?
Well, like the little bowl thing.
Yeah. Okay? So pretend that Pac-Man would be the what? What is he? The enzyme. And then what he eats would be the substrate, right? Okay, because that is going to go in his belly and become something else, right? Okay? But keep this in mind because the next thing we're going to talk about, what else does he eat? And what do they do to him? So he kind of, right, because it's almost like he would die, right, if he had too many ghosts, right? All right, so keep this in mind for the next thing. All right, so one last thing before we move on. What's an enzyme? 2 words. What's an enzyme? So which macromolecule is it?
Protein.
Proteins, and it's a what? Begins with a C. Catalyst, a protein catalyst, because it does what? Speeds things up, speeds up the reactions by
Lowering activation.
lowering the activation energy. Once again, okay, what does it mean to say they're specific? Like a lock and key, and reusable? Very good. And why are they important? Okay, they help digest sugars, but they really help in the, okay. All right, so the next little bit, this is pretty short and sweet, but you also need to know this, and this is super, super important. So an enzyme's job is determined by its shape. So not every enzyme is going to look the same, okay? So what happens if we change the shape of an enzyme? Okay, so you see the first one, do they fit? Okay, what about the second one? No, okay? So what happens here is what we call denaturing, okay? So an enzyme changes its shape and it will no longer work. So if the enzyme shape changes, the substrate's not going to fit, so therefore it is not going to work. That is called denaturing. Make sure we remember that word, it's super important. So because the shape of the enzyme changes, the substrate and the enzyme aren't going to fit like a what? Lock and key, okay? So therefore, it will not work. Enzymes can be denatured by the following things: extreme pH and extreme temperatures. Okay, salinity content also plays a role, but these are the 2 major ones: extreme pH or extreme temperature. So if we were to add a whole lot of heat to an enzyme, it will make it change its shape. Okay, extreme temperatures. Something else we need to be familiar with, and I'm sure we've seen this before, what is this? The pH scale, right? Okay, so down towards the bottom, over here, acidic, over here, base, basic, okay, or alkaline is another way to look at it. What's neutral? Seven, neutral. Okay, so 1 to before 7, or 0 to before 7 is acidic. Anything after 7 is basic. Okay? Something else we should know, has anybody ever heard of a buffer? You heard the word before? Okay. Buffers are super important because they act as a neutralizer. What does it mean to neutralize something? To equal it out, to even it out. So if something's too basic or if something's too acidic, we want it to be equal, neutralized. So that's what a buffer does. Okay? All right, one of the last pieces here. So each enzyme has conditions that it works best under. The word we use for this is optimum. So optimum means best. Okay? So in order to find the optimum pH or temperature, we have to do 2 things. We have to find the place with the highest peak. Then we go down and read whatever is on the x-axis. Whatever's on the x-axis, that's the number. Sometimes it might not be an exact number, so we just need to guesstimate, okay? So, to the top, drop it, and read the x-axis. Everybody with me? All right, what's graphed on the x-axis here? The what?
You said what graph of the X?
Yep, what's graphed on the x-axis? Which one is the x-axis, horizontal or vertical? Horizontal. So what is that down there? Those numbers, what are they representing? The pH. What's graphed on the y-axis? The rate of reaction. So looking at this, what is the optimum pH for catalase? 7. That's it. Is that hard? All right. Are you sure? To the top, drop it down. All right. So here are just some other examples. So the optimal pH for pepsin, which is a stomach enzyme, would be what? Around 2. And then what about for trypsin, which is an intestinal enzyme? About 8. Okay. Here's another one. Now, this one's not exact, so give me for a human.
36, 35, maybe.
Okay, I'd say somewhere about there. Yep, 35, 36. And what about this guy?
51.
A thermophile. About 50, 51. Okay, so give or take. If you're close, I would never mark it wrong. But you gotta get close. All right, so real quick, and then you are going to work on what I have assigned for today. Once again, if we don't finish it today, you can work on some tonight, but know that you'll have a little bit of time tomorrow. So, what does it mean to denature an enzyme?
To
Change its shape. Does it work? No, because it doesn't fit like a what anymore? Lock and key. Perfect. And optimum means what? Best. That's it. Keywords.